Hi, so my name is Ryan Morris, and first of all, I'd just like to thank everyone for giving me the opportunity to come talk about my research. Interacting with the scientific community is probably one of the most fun things that we get to do. So my project is titled Developing Luminescent Probes for Imaging Mitochondria in Fixed Tissues. So high energy demanding tissues, such as muscle tissues, are particularly reliant on mitochondria to provide energy in the form of ATP. Mitochondria also provide a number of other functions, such as the regulation of ion homeostasis and the production of reactive oxygen species, just to name a couple. So just a quick overview of the structure of mitochondria. So mitochondria are quite unique amongst organelles because they have two phospholipid membranes as opposed to one. So first of all, you have the outer membrane, followed by an aqueous intermembrane space, and then there's the inner membrane. And in the inner membrane, that's where ATP is formed via oxidative phosphorylation. So in the process of oxidative phosphorylation, proton pumps pump positively charged hydronium ions out of the out of the mitochondria, and this creates a negative membrane potential. So remember that negative membrane potential because that will come up shortly. So changes in mitochondrial morphology are part of the pathophysiology of a number of different diseases. So cancer, cardiac disorders, diabetes, and neuromuscular disorders, just to name a few. So this is a really good example. I'll see if I can get this to work. Okay, I cannot figure out this thing. All right, so these dark patches here are mitochondria. So this is a sample taken from a patient with myositis, which is a neuromuscular disorder. And you can see there's these really irregularly shaped mitochondria. There's one, for example. This is really difficult to use. And that irregular shape can lead to inefficient production of ATP, which can lead to muscle weakness and difficulty in moving. So this is a really good example of how imaging mitochondria can give us a good indication of disease presence as well as progression. So what forms of imaging do we have available for imaging mitochondria? So the two predominant methods that we use are electron microscopy and fluorescence microscopy. Electron microscopy has really high resolution. You can see this image on the left here. That's an individual mitochondrion, and you're capable of seeing the submitochondrial structures. So it's really great to have that high resolution. However, that high resolution comes at a great cost. Like literally, it's quite expensive. Electron microscopes are very, very expensive, as opposed to fluorescence microscopes, which are comparatively a lot cheaper and much more readily available. Electron microscopy, also the techniques that we use to produce contrast are quite harsh, and that can lead to the introduction of artifacts. The biggest drawback with using electron microscopy for imaging mitochondria is the stains that we use to produce contrast in electron microscopy stain everything indiscriminately. So when it comes to imaging mitochondria, particularly in tissue samples, where it's a far more complex environment, you really need selectivity so that you can visualize mitochondria. So for that, we usually rely on fluorescence microscopy, which we use probes that are designed in such a way that they selectively stain mitochondria. So in this image here, you can see each one of these red patches are mitochondria and just mitochondria. So they're selectively stained and you only see the mitochondria. So when it comes to imaging tissue samples, fluorescence microscopy is what we rely on. So what's fixed imaging? I imagine most of you have heard of fixation before. The goal of fixation is to preserve your sample in as lifelike state as possible. So there are a number of different methods that we use to do this. There's both physical and chemical methods, the most common of which is to use formaldehyde and paraffin. So in fixation, biological functions cease and your sample is fixed in place. So So nothing's moving anymore, so that produces a high stability of your sample and improved optical transparency, which allows us to produce really high quality images. It's also often just more practical to fix your sample. So say you did 100 specimens, 100 samples from a single specimen. If you wanted to image them while they were live, it could be quite difficult with that many samples. So you'd have to take into consideration pH, temperature, and a number of other factors. So often it's just most logistically feasible to fix your samples and then you can analyze them when you're ready. So what probes do we currently have available for imaging mitochondria? So conventional probes for imaging mitochondria are organic. So there are a number of organic probes available, including rhodamine 123 and JC1, just to name a couple. The issue with existing organic probes is that most of them are designed in such a way that they're optimized for staining cell samples, and the selectivity in cell samples doesn't necessarily translate to the more complex tissue environment. However, the most commonly used organic probes are mitotrackers, and mitotrackers have the benefit of being able to stain both cell and tissue samples. However, it comes with one huge caveat that you're only able to stain live samples. So all these probes here, you can see they're all positively charged. So they're able to isolate in the mitochondria by exploiting that negative membrane potential that I mentioned earlier. So the positive charge draws it into mitochondria. However, if you've fixed your sample, biological functions have ceased. So proton pumps are no longer pumping, and you no longer have that negative membrane potential, and you're unable to localize in mitochondria. So recently, inorganic probes have, a number of inorganic probes have been developed for mitochondrial imaging as well. And iridium is really the star of the show when it comes to inorganic probes. There are iridium probes that are capable of staining both live and fixed samples. However, as with the organic probes, most of the probes that we have available are optimized for staining cells and not tissue samples. So there are also a number of other metals available, including rhenium and ruthenium, just to name a few. So just a quick overview, with the organic probes, we've got probes that can stain cells and tissues, but only in live samples. And with the inorganic probes, we've got probes that can stain live or fixed samples, but only in cells and not tissues. So you may be asking, do we currently have any probes available for staining fixed tissue samples? Well, yes, we kind of do. So up until recently, we've had to rely on immunochemical methods for labeling fixed tissue samples. So that's where we use antibodies that are bound to fluorophores, and the antibodies selectively bind to proteins in the mitochondria. The issue with using this method is that these antibodies are really, really expensive. The process is quite time-consuming, so it can take up to several days to get the sample ready for imaging. And there are several steps involved in that, each of which are prone to artifact formation, plus we're limited by the availability of these antibodies. Now I say up until recently, because as of 2018, there is currently one small molecule probe that is available for staining fixed tissue samples. And that's great because small molecule probes are significantly cheaper to produce. The incubation period is a lot faster. It's only about half an hour as opposed to several days with the antibodies. And this one's commercially available already under the name Irresolve Mito. So that's great that we've got one small molecule probe available. However, only having one probe is quite limiting with our options. So say you've got autofluorescence from your sample that's masking emission from your probe. Given that there's only one small molecule probe available, if we would then have to resort back to using the expensive antibodies. So if we could develop a range of alternative probes with varying photophysical properties, we would no longer have to rely on the expensive antibodies. So you may be wondering, why is there only one small molecule probe available at the moment? Well, it was seemingly discovered purely by chance. So the initial investigation where it was synthesized was a project aiming to develop more probes for live cell imaging. And then of all the probes that they designed in that investigation, they also stained fixed tissue samples. And irresolved mito was found to stain in both live and fixed samples. So this compatibility with both live and fixed samples suggests that its method of localization is independent of the mitochondrial membrane potential. But given that it was discovered by chance, we don't actually know what structural elements are leading to that selectivity. So that brings me to my project, where my aim is to investigate the structural requirements for fixed tissue stains, and in doing so, develop a library of fixed tissue probes with varying photophysical properties. So there are a number of structural elements that I would like to investigate. So there's the cyclometylating ligands, these ones. Sorry, this is really difficult to use. There's the ancillary ligands, this one. There's the metal center, the charge of the complex, as well as the lipophilicity. So these are elements that I aim to investigate. So how do I plan on doing this? So starting with the base compound, being the compound that we know stains fixed tissue samples, I'm going to systematically alter that compound and investigate the effect that changing those elements has on biological selectivity. So I will synthesize a range of compounds, each varying one element, then stain biological samples, then perform statistical analysis, and based on results, come up with further modifications. So I won't be doing that in a linear fashion. So for efficient use of time, I'll be synthesizing multiple complexes concurrently and then doing staining at the same time. So what sort of structural investigations do I want to carry out? So first of all, I want to see the effect of the metal center. So is it necessary for it to be iridium, or can I use alternative metals to perform the same function? So as I mentioned, rhenium and ruthenium are two good candidates, as we've already shown that we can localize them in mitochondria. I want to explore the effect of the charge. So does it need to be cationic? Given that there's already evidence that it can localize in mitochondria independent of the mitochondrial membrane potential, it may be possible to produce neutral analogs that can perform the same function. I want to explore the effect of conjugation. So I can do this by altering either the cyclometylating ligand or the ancillary ligand. So I can either increase or decrease the conjugation of either of them. Another thing I want to explore is the effect of the tetrazole, the four-membered heterocycle. Does it need to be a tetrazole, or can I substitute it for other azoles, for example, a triazole, as I've shown there? This would be particularly interesting because that opens up a whole range of things that I can explore. So in summary, mitochondrial imaging is essential in the analysis of disease progression and presence. We currently only have one small molecule probe available for staining mitochondria in fixed tissue samples, and my aim is to investigate the structure for fixed tissue probes, and in doing so, develop a library of alternative probes and advance our knowledge in mitochondrial probe design. Big thanks to everyone on this list. And yeah, any questions?